Thank you, Kasturi, and others, and thank you for inviting me to give a talk in this platform. I was very impressed to hear about the initiatives of this platform, the center, and what that we are trying to do. Very exciting, I'm sure, and these are very important, and these such initiatives will have a lot of impact. So I am a little pressed for time today, so that's why without further ado, I would just like to launch straight into my talk, and then at the end of the talk, I hope to take a few questions as the time permits, so that and then I will leave with my email address so that people, I'll be happy if people can contact me on email later on as well if they have any questions or if they have any interest in the kind of work we do, would like to engage more scientifically in this kind of things. So let me share my screen in the beginning. So I would actually like to share the whole screen. Share. Perfect. So is my screen in, I mean, the slides are in slideshow mode?
Yeah, they are visible.
And they are changing, right?
Yes.
Great. So thank you very much. And as I said, so I will straight away launch into my uh, hide it. Okay, doesn't matter. So I will launch into the talk that I am promised to give, which is our latest insights into single cell transcriptomics of gingival oral cancer. Now, before I do talk about it, and let me give you a little information on, at least to try to give some information on the whole range of people who actually have been I've been working with, and who all made it possible for this kind of work to happen. So first and foremost, I have my three closest associates who are actually pillars and who are the driving forces of this initiative. And one of them is Dr. Silarine Kurkalang. She actually was my postdoc for some a long time, helped me with the initiation of this single cell transcriptomics in NIBMG and various ways. And now she is no longer with us. She has joined the University of Chicago and working on single cell genomics and using in a CAR T cell therapy lab. Sumitavo is my senior most PhD student who has just completed his PhD and is scheduled to appear for his public defense. He is the first person who started with, along with Celerine, on this single cell initiative on oral cancer. Arunima is our IPhD student who later joined me for PhD and is carrying forward this single cell transcriptomics work. There are various other people in different capacities who actually helped us in very different kinds of ways. Most important of them is our clinician collaborator, Dr. Sandeep Bose from R. Ahmed Dental College Hospital in Kolkata. It's the largest dental college and hospital of the government, and actually sees a lot of patients. And he's a fantastic and exciting pathologist to work with. And Professor Parthamati Majumdar, who was the initiator, kind of pushed us to get into this field, and retains a very active scientific engagement in this kind of newer kind of genomics analysis. Also, I had the backing of the large phenomenal genomics facility of NIBMG, which I also incidentally which I manage. And this wouldn't have been possible without the existence of such a genomics facility, which made entire work possible. Now, as we know that cancer is an evolutionary process, and right from the beginning, from the fertilized cell to egg to the ultimate somatic cell, and subsequent rounds of cell division in the somatic tissue, there are multiple introduction of these replication errors, which gets hard coded in the genome, and which ultimately results in a mutation, which is subsequently harbored in the progeny or the daughter cells. Fortunately, most of the times, as we know, that these mutations are selection neutral. They do not confer much change in the phenotypic property of the cell, but only rarely a mutation starts to give a selection advantage to the cell. The cell expands, and there are minor winners in these kind of, so it expands and it replicates a little better than other cells in the same tissue in its surroundings. And examples of these minor winners of these competitions are like many of us harbor moles on our skin. So these are such examples. But rarely does a mutation happens in a certain gene which disrupts the phenotypic property of the cell. The cell starts replicating uncontrollably and then becomes invasive. And that's the birth of the tumor, invasive tumor. And then this tumor then clonally expands and then ultimately metastasizes. If there is treatment, then there are treatment resistant clones that develop, and all these happen due to mutation. So we know now cancer is a genomic disease. Fortunately, the vast alteration, 90% of these mutations that happen are somatic, and only 10% of them happen in germline. And these somatic mutations, which are the most predominant cause of various types of cancer, has been last decade, there has been intense searches to find out all these genes that are changed by these mutations and which result in tumorigenesis. So these mutations are driver mutations, driver genes, just like the driver in a bus, the single person who controls, and the rest of the mutations are passenger mutations. So over the last decade, disruptive changes in the sequencing technology and our ability to sequence the tumor and paired adjacent normal tissue from cancer patients have resulted in cataloging of these somatic alterations that drive cancer, various kinds of cancer, resulting in which was made possible by this phenomenal international consortium called the International Cancer Genome Consortium, which sequenced about 25,000 genomes at a deep level and identified for most major cancer types, identified these mutations and cataloged them. India was a founding member, and NIBMG spearheaded this initiative from India. We worked on gingivobuccal oral cancer, which resulted in our present-day understanding of the most of the genomic alterations and transcriptomic alterations that drive this cancer in India, primarily caused by chewing of tobacco. Now, beyond this, the point is, so when this was happening and this took shape, we also realized, and this is a global understanding that developed, was that we are looking at a large number of cells, and the cells, when we look at gene expression, the different cells have different kinds of gene expression. All cells harbor 20,000 genes, but only a few of them are expressed in various cell types. So how understanding these cells are important, because the cell is actually the effector of the genotype to the phenotype. So, and that's why understanding how each of these cells express the genes, which genes they express at what levels, is key to our understanding of the role of these genetic variants and the genome in affecting a health phenotype like cancer. So this was how the Human Cell Atlas mission was set up. It's still continuing. There's a lot of progress happened because of, again, very large developments and leaps in technology in our ability to sequence individual cells. And the idea is to create a periodic table of cells, understanding each and every cell, how they function, how they express the genes. So there are various methods by which we do this kind of single cell sequencing, but the most important, the most used and stable, one of the most stable technologies that we use in NIBMG are using the 10x Genomics Chromium platforms, where we make a cell suspension if it is a solid tissue, and then using microfluidics in the 10x Genomics platform, we actually create water droplets in oil, where in each of these droplets, one cell gets harbored, along with the oligonucleotide harbored on beads, and then enzymes and other reagents required for reverse transcription and PCR. So within each droplet, the cells are dissolved and the RNA is liberated, and the RNA binds to the surface of these beads, which have these oligonucleotides. Reverse transcription happens, and then PCR happens, and then these PCRs then introduce these kind of adapters on the cDNA that are generated, and these are used for single cell sequencing. So there are two kinds, and then we take these prepared sequencing libraries. People who are accustomed to next generation sequencing would understand that, and then we perform sequencing, which is no different from bulk sequencing. But there are two different things that actually differentiates and enables us to decipher this data back to the sequencing signal that we receive from each cell. On one, we have two different kind of barcodes in the oligonucleotides. One is a cellular barcode. So this barcode is specific for each bid that, and in the droplet, there is one bid and one cell. So all the cDNA that comes from a particular cell gets encoded by only one type of barcode, and that gets copied and amplified and sequenced. But when we sequence, because there is this amplification, it's very difficult to draw quantitative inferences because of amplification bias and so forth. So what we to correct that, what we have also done is there are something known as unique molecular identifiers. These are again short random sequences. They are present in excess numbers in our reactions. So each cDNA ultimately for each transcript we can eliminate the PCR amplifications that have happened and count to how many transcripts for each gene in a cell we had. So using these two kind of barcodes, we are able to decipher the single cell gene expression data for each sample. So, but then these kind of approaches, although now quite established, we are doing a lot of it. I will talk about it, but there are severe challenges. We do not know for a given tissue what are the large number of cells and what are their frequency ranges. These are unknown most of the times, and there is a broad frequency range. Now, each tissue has its own structure, cell morphology, and hence there are different challenges for dissociating the cells without losing any significantly the viability of the cells. Then also there are these issues that there are rare unique cell populations in tissues and particular conditions where we need to identify them, but we do not know whether our methods capture them efficiently. And finally, we need to, whatever inference we draw, we need to harmonize with other non-molecular classifications. A big example is cancer. So what we did was, when we started working on gingivobuccal oral cancer, after our bulk genomic approaches, when we launched here, we actually tied up with this dental hospital, which is very close to us, about 30 kilometers from our institute. And we took freshly dissected biopsy samples of treatment naive early stage oral cancer patients. And then we bring them over to our institute using a very rapid transport, and I will tell about it a little bit later. And what we did was we sequenced the primary tumor of, we profiled about 52,000 cells from 12 patients of OSCCGB. And we used this 10x Genomics Chromium to generate the single cell libraries and then sequence them on NovaSeq 6000. And we have our own pipelines combining with what is publicly available. And we ultimately have done the analysis. So when we generated the single cell data, we needed to understand how good this data is. I mean, whether does it reflect the gene expression heterogeneity that we normally see in using bulk RNA-seq approaches. So for these oral gingivobuccal oral cancer, we have actually sequenced a large number of patients' tumor and not just a normal tissue in patients in ICGC, and which is published. So we compared the number of genes that are expressed, and we find that compared to the bulk data, so these are the patients that we sequenced, all express similar number of genes. Then we also compared whether the average expression of common genes between the bulk data set and our single cell transcriptomic data set, and also average gene expression of about little more than 1,000 genes, which are upregulated in the bulk tumor and the bulk sequencing data they obtained versus normal, and compare it with our single cell data. In both the cases, we found that there is substantial correlation, which told us that we are able to capture most of the heterogeneity of expression that we are seeing in bulk in our single cell data. So then after this data is generated, I won't go into nitty-gritty details. We generally, this is a multi-dimensional, high-dimension data and complex data. So we use dimensionality reduction techniques, which is like the one that we use right now, and most of the community uses is UMAP, where these each dot here is a cell, and each and depending on the expression of these genes in each of these cell, they are closely spaced or they are far apart. If two cells are far apart, they have a diverse gene expression program. And if they are closely spaced here, that means they have very similar gene expression pattern. So when we did that and we looked at the epithelial cells, because this is an oral cancer, initially when we were doing that, we found that very interestingly, one of the patient's cells will be clustering away from the all other samples. So we were very curious, why is that? What is happening? And then we went back to the clinician and then we found that by mistake, when initially these samples were being given, one tumor of which is known as oral verrucous carcinoma was included, which should not have been included. So verrucous carcinoma is not so aggressive and it is a less malignant form of the tumor, and that is not treated as OSCCGB. So you can, but what made us feel good was, using the method, we can actually differentiate the different, the cells that are coming, epithelial cells that are coming from this oral verrucous carcinoma from all other samples of oral cancer. So that told us that, we are, this kind of method and technique is very robust and very sensitive. It can actually differentiate even between the subtypes of the tumor. So then we moved into classifying each of these cells into what cell types they are. Now here we go back to biology, understand what are the different cell types and what are the markers of each of these cell types. And then we go back to each cell's gene expression data, which is a cell by gene matrix, and we find out which of these marker genes are expressed in these cells. And based on that, each cluster we can classify or characterize as which cell type they are. So here on these 12 OSCCGB samples, we found that there are 7 major cell types and there are many minor cell types. We could identify the epithelial cells, fibroblast, endothelial cells, myeloid cells, mast cells, and T cells and B cells. So using various kind of markers, and as you can see on the right panel, yes, please. So then the next question came, if you go back to this, we have these epithelial cells. Now, the malignant cells will reside in these epithelial cells, but whereas all malignant cells are epithelial cells, all epithelial cells are not malignant cells. So we had to differentiate and identify which are the malignant cells in these epithelial cells. Now, there are methods that are in use where one uses the gene expression data of each cell and then uses a baseline generated from non-epithelial cells as a baseline. One compares and creates an inferred copy number, assuming that if you have more gene expression for a particular gene, then the copy number of that gene is increased. And we know that in malignant cells, there is a large-scale copy number alterations compared to the non-malignant cells. So this kind of approach where we infer a copy number from the gene expression data is used in a method called infer-CNV, which is mostly used by the community until we generated our data. So we decided to use that and we could identify cells using that. But then we are not satisfied because these are not actually the methods. We found that, it's an indirect way of looking. We are generating expression data. Why not use the expression data directly instead of inferring a copy number and see if we can develop a more sensitive method? So what we did was, here Professor Majumdar played a role. He came out with this concept of this, using Euclidean distance, using the gene expression data. And we found that this kind of distribution has 2 nodes. So one mode is here and the other is here. And so we found that there is an anti-node and that kind of separates out them. So on the right would be the malignant cells, on the left would be the non-malignant cells. So using this approach, which we called a distance profiling method, we were able to classify malignant cells versus non-malignant cells. And when we compared the classification we did with the established method of infer-CNV, we actually used gene expression programs of two kind of pathways, biological processes. One is apoptosis, the other is DNA repair. And we found that the cells classified as malignant using our method actually shows a vast reduction in apoptosis and DNA repair pathways compared to the cells expressed as identified as malignant by the infer-CNV. So we realized that our method was actually doing a better job in classifying the malignant cells. So once we validated it, then we decided to use this method and classified our cells into epithelial cells into malignant and non-malignant cells. Now, when we go back to our non-malignant cells, which are like non-malignant epithelial cells and various immune cells and fibroblasts, we've found that these cells actually cluster by cell type. So the clusters were predominantly by cell type and not by the tumor of origin or the sample. So this shows that between individuals, there is not much heterogeneity in expression of genes in these non-malignant cells. And when we looked at this distribution, we found that there is a varying distribution of non-malignant cells in each of the tumors. But when we looked at the malignant cells, the situation changes. What we found was that most of the clusters generated from malignant cells were tumor specific. That means for each, they were patient specific. For most of them, each patient contributed largely to a single cluster, and that was different from another patient. So there is a lot of interpatient heterogeneity of gene expression in malignant cells. But however, we also found that there were a small number of malignant cells which also were shared between multiple patients. So with both kinds, so that actually the expression data of malignant cells are complex, and we found that this is kind of, there's a lot of heterogeneity in that. So again, understanding using these kind of methods is the only way to decipher this level of heterogeneity in gene expression in the tumors. We looked at each of these malignant cell clusters, and to cut a long story short, we found that each cluster had its own typical gene expression pattern. We could find that these malignant cell clusters were divided into two groups, whereas group 2 consisted of patients from all 12 patients from but clusters in group 1 predominantly were limited to only 2 patients. And when we went back and found that these 2 portions also had a concomitant precancerous state, which is known as oral submucous fibrosis. Now, oral submucous fibrosis is a very painful precancer condition, which is very common in India, associated with tobacco and areca nut combinations. And some of them develop into oral cancer. So these 2 patients actually developed oral cancer from OSMF. And they had some malignant cells which are typically present in them and not in other OSCC patients. Now, in genomics, we always have this, and most of biological comparisons, we always compare the initial versus the final state. So we actually dichotomize our readouts and try to compare initial versus final. But during the way the cells operate in nature and in our system is that there is more like a dynamic continuum between different kinds of cells and different cell states as opposed to a dichotomy. So just like, if you just look at this initial and the final pictures of a long jump runner, you will not be able to make out. But actually what matters most are each of the steps between these two initial and final states, which ultimately results in dictating how much distance actually the long jumper is able to achieve. So the final destination or the final state is dependent upon the entire process of transition. Now, without standard bulk genomic approaches, we are not able to understand these kind of changes or dynamic continuum within the cells in a particular biological tissue. In single cell transcriptomics, we are able to do that because we generally sequence a large number of cells. And what happens are, if there are multiple different kind of cell states, intermediate cell states, we are able to sequence each of them. And then we can actually do something known as a pseudotime trajectory analysis to create and understand how these cells between which states they evolve into which ones. So here, like EMT is a very well-known kind of very well-known kind of event in cancer. I don't have to talk much about EMT, but what we found where there are some cluster of malignant cells which are in partial EMT. That means they are just in transition from epithelial to mesenchymal transition. So we found using there are partial EMT has been described earlier, and we used the known expression of genes in partial EMT, and we found that there were about 6 to 7 clusters of malignant cells who expresses the gene expressions of associated with partial EMT. Now, when we compared, so these clusters, when we compared their expression correlation with gene expression of known EMT associated pathways, we found that there is a very high correlation in all the clusters except one, which is called the cluster 15. And interestingly, when we go back, we find that this cluster 15 is contributed by cells from only those 2 patients who had OSMF associated with OSCC. So this cluster 15 is interesting. They probably have a difference in the EMT expression, and it requires a deeper investigation into what's going on right there. So partial EMT is very important because partial EMT plays a very important role in the local tissue invasion, in collective migration, or in generation ultimately of circulating tumor cells. And ultimately that results in loco-regional and distance metastasis. Actually, now we are trying to collaborate with clinicians who have a very deep focus on circulating tumor cells in oral cancer. And we are trying to see whether we can actually track which cells from the tumor actually go on and generate as circulating tumor cells in the peripheral blood of these patients, and so that we can see whether they have a heightened particular partial EMT program that enables them to do that. So we also looked into various kinds, all the other malignant cells, and we found that interestingly, many of the malignant cells from these patients expressed 2 different kind of gene expression programs. One was expression of fetal expression of fetal genes. So these fetal gene expression signatures are known in other cancer types, and we found that many of the cell clusters of malignant cells actually expresses the squamous epithelial cells signature, fetal lung squamous epithelial signature, and fetal squamous epithelial signature. So this was the fetal gene expression signature, and the red shows that these clusters are actually expressing them in a higher proportion. So there are cells which express partial EMT signatures, there are cells which express fetal genes, and then we also found there are 3rd type of malignant cell clusters which express their germ cell-like gene expression signatures. Essentially, most of these are cancer testis gene family members, and these are also known to be expressed by cancer cells, some cancer cells. So we found that all these 3 gene expression programs are there. And what is more important, we found that they are mutually exclusive. So the class cells either express PEMT or fetal expression signatures or cancer testis gene signatures. So then we did this pseudotime analysis, and we found that interestingly, what might be happening is that one end is our partial EMT cells, and on the other end of the evolution are these cells expressing fetal genes and cells expressing germ cell or cancer testis genes. So might be that the PEMT cells are then differentiating, or vice versa, into genes which cells which express these fetal genes, or the cells which express these germ cell-like signatures. We also looked into a group of cells known as slow cycling cells, and we found that these are very limitedly described in oral cancer. The slow cycling cells have this ability to self-renew. They have bare stem-like properties, and we found that we identified 2 clusters, cluster 14 and 16 in malignant cells, which express this slow cycling cell signatures. Now, earlier in classical biology, these slow cycling cells have been identified and their cell cycle stages have been identified by their ability to intake dyes. And but what we found was when we use these gene expression signatures, and it has been reported that they are all in G0, G1 stage. But when we looked at this cluster 14 and 16, and because we are able to use the gene expression signatures of all the 20,000 genes to classify them, we found that actually these slow cycling cells have different. So although most of them are in G0, G1 phase, but there are substantial number of the cells which are actually in S phase or G2M phase. We also found that these slow cycling cells form separate clusters like what is shown, and these express NEAT1, MALAT1, and various other genes which are very important in promoting tumor growth and metastasis. So finally, we actually identified two clusters, cluster 14 and 16, we express slow cycling cells. But interestingly, we found that cluster 14 also expresses partial EMT, whereas cluster 16 expresses fetal gene signatures. And this NEAT1, MALAT1, and these PLCG2 and LAMC2 are expressed by both these clusters. So these two clusters might be very important, and we are actually now homing in on doing large number of patients where we are investigating these two clusters, and we are also trying to investigate the role of OSMF-associated OSCC. We also, I will skip this slide. So we moved into the non-malignant cells. The non-malignant cells are large numbers in nature, and most of them we found that the T cell, actually there are large number of T cells present. And we found that in the T cells, there are various kinds of T cell states, those that show exhaustion programs, cells that show cytotoxic programs, and cells that show both. And particularly, we found that there were a group of T cells which are double negative, that means CD4 negative, CD8 negative. So our understanding is, so we homed in on that, and we found that these T cells actually, the double negative T cells actually also express PLCG2, which is phospholipase C gamma 2. And this is enriched in this double negative T cells. And we found that these double negative T cells, we did a trajectory analysis, and we found that these double negative T cells probably might be generated by loss of the CD8 antigen from the CD8 positive T cells. That is a hypothesis based on our single cell data, and we are now getting into downstream biology, in vitro cell experiments to identify whether actually that is true or not, and what are the role of these double negative T cells, because we feel that these are important for tumor progression and ultimately response to treatment. So we also looked into macrophages, and to cut a long story short, we found that the macrophages can be classified into M1 and M2. M1 are pro-inflammatory, M2 are anti-inflammatory. And we, again, to continue that continuous transition theory, we found that there are a large number of macrophages which show a transition from M1 to M2. We also looked into cancer, the fibroblasts, and we found that we could differentiate the fibroblasts into cancer-associated CAF and MyCAF. And then we found the subpopulation of fibroblasts which are like ICAF, and we again found that there is this undifferentiated fibroblast actually move into either using pseudotime trajectory analysis. We found they either evolve into CAF or they evolve into MyCAF. So the fibroblast, there is a lot of interesting thing happening in the fibroblast, and we would like to actually looking deeper into it in larger number of samples and also on in vitro cell experiments. So finally, we actually, in the initial data that we generated and analyzed, we found that there is extensive cellular diversity in OSCCGB. Both intra and inter-tumor heterogeneity exists. Malignant cell populations possess patient-specific transcriptional programs, and there are some shared programs as well. OSCCGB-associated OSMF tumors actually exhibit expression profile which are distinct from non-OSMF tumors. And we found that there are two dominant gene expression programs, partial EMT and fetal cellular reprogramming, that is happening within these tumors. Non-malignant cells, we found that double negative T cells are a very interesting set to work on. And in future, we will be investigating more on this M1, M2 transitions and these evolutions within the cancer-associated fibroblasts. So this is where I will end. Our lab also works on multiple other single cell programs. One is in placenta and different gestational ages of placenta, different gestational ages, to understand what are the cell type diversity changes that happen between preterm and term deliveries. We are also participating in this Asian Immune Diversity Atlas to characterize the healthy immune state of Asians, which are underrepresented in global databases, using single-cell transcriptomic on peripheral blood cells. And in that, our focused question is also understanding how single nucleotide polymorphisms actually control gene expression in a cell type and cell state specific manner. And we have a large lab where we work on oral cancer, genomics of prematurity, and on gene regulation. And in addition to our research, we also have a lot of fun on the weekends doing walk tours in Kolkata and eating street food. Thank you very much. I would stop here, and if there are any questions, I would be happy to take.
Thank you, Dr. Maitra, for this very comprehensive talk. There is one question from Gauri. Could you please repeat the distance profiling method?
Okay, so what we have done here, I mean, the details are there in the paper, in our cancer science paper that has come up in a supplement. We have provided in the supplementary the details of it. So what we have done is we have calculated Euclidean distance of gene expression in each of the epithelial cells. And then we have plotted the density of those, and we find that there are two modes. So the one on the right that I showed is where they have very high gene expression distances, and that is from we hypothesize that is from the malignant cells, whereas the mode on the left, which has lower Euclidean distance, is from non-malignant cells. So actually we are trying to use that, and we see there is an anti-mode. So we use that anti-mode with some confidence interval to classify what are malignant cells. Now, this is what we hypothesized, but when we actually tested using these multiple gene expression programs, in particular, I showed this apoptosis and DNA repair, which are down-regulated in most of the cancers. So we found that the malignant cells classified by this method actually have very low or lower expression of these two programs compared to cells classified malignant by other methods.
Great. So in the interest of time, okay, I see another question has come up. Thank you, Dr. Maitra, for a very informative talk, says Ruksar. My question is a bit towards the clinical side. What are the chances of relapse of oral cancer after surgery and radiotherapy?
That is $1 million question. Actually, we are using bulk sequencing approaches to there is a study, and we are right now analyzing the data where we have taken oral cancer patients with the primary tumor and also tumors. So we have patients in two groups: patients who have recurred after surgery within 2 years, and patients who have not recurred within 2 years. And we have done bulk transcriptomics and whole genome sequencing. We are analyzing that data, but would be happy to actually. So once we learn more extensively, right now we have completed the single cell transcriptomics of about 30 patients with and without OSMF on OCC, and we are in the process of doing a deep analysis of that data. So whatever signatures we find, and I didn't mention that, we are now going to go back into all this bulk data that we generate, because it's much easier and cheaper to generate bulk data compared to single cell data. We would like to use these expression signatures into the bulk data and then see how they correlate with this treatment recurrence and disease-free survival period in the bulk patients where we have the bulk data, which is much more in number, and those there the patients have a good follow-up data that we will be able to use.
Great. Sounds very promising, Dr. Maitra. Thank you so much for your time. I understand that you must be extremely busy with all your research, academic duties, as well as with being the associate director of the center. But thank you anyway for honoring us with your presence and your talk. I request all the participants of the webinar today to get in touch with you if they have any queries.
Sure.
And we would love to host you physically at our center anytime. So if you're in Hyderabad, do come over.
Sure, we'll do that. Thank you
Yeah.
very much.
Thank you very
Bye-bye.
much. Bye.